Our presentation today is on invertebrate evolution. It's by me, Xander Fair, and my partner, Isaac Simmons. So first of all, we're starting off with what is invertebrate evolution. Invertebrates are species of animal that neither possess nor develop a vertebral column, or what we refer to as a backbone or spine. Invertebrates compose around 95% of the animal species and have been around for about 550 million years and have came a long way since then, both evolving and changing. The three main things that developed during their evolution were symmetry, specialization, and cephalization.
Okay, and one of the first traits to evolve was multicellularity, and it was one that made them very adaptable because it allowed different cells to branch out and focus on specific tasks. And these cells would become more efficient as a result. And a good early example of this would be a sponge.
Alright, after multicellularity came the evolution of tissues, which would further lead to the development of organs and organ systems. Invertebrates developed tissues from two embryonic cell layers, the ectoderm, which is the outer cell layer, and the endoderm, the inner cell layer. Examples of this are cnidarians.
And the next is radial symmetry. There's two different types of symmetry. There's bilateral, which is two identical halves, or radial, which is identical sections. Radial, like circle, is how I think about it. And radial symmetry evolved first, and it has sensory receptors in every direction, which allows the organism to react to food or defense in all directions. But the problem with it is that they don't have very precise movement as a result of it. And a good example of this would be a coral polyp.
Yep. And then cephalization, which is another stage of evolution. This particularly focused on the nerve tissue at the interior end of the body, which would form a head and a brain. This would allow this basically allows central control of the entire organism, which acts as a brain and well actually is the first steps in the evolution of the brain. An example of these would be flatworms.
The other type of symmetry, like I said, was bilateral, which is two halves. And what this really allowed was for organisms to differentiate forwards from backwards and left from right, which allowed for more precise and controlled movements. Which a real good example that you could see is a spider because it has four legs and a line down the middle really splits it into two halves.
Alright, and then now we have the mesoderm, which is the evolution of a new cell layer, which would allow animals to develop new types of tissues and develop muscle. This third layer of cells is between the ectoderm and the endoderm, and it's evolved from the ancestors of flatworms. And you see in this picture down below, it's right in between. Yep.
Okay, and the next is a complete digestive system, which early invertebrates had a single opening for their mouth and anus. Eventually, separate openings evolved, and this allowed organisms to continue eating while digesting waste, and for more efficient digesting, which would eventually lead into evolving individual digestive organs. And a good example of this is earthworms and grasshoppers.
Alright, so the pseudocolumn, which originally evolved from the ancestors of roundworms, is a partial body cavity that is filled with fluid, and it both cushions the organs and allows them to develop, which is basically a hydrostatic skeleton. The column, it's a full filled body cavity and is completely enclosed by the mesoderm. And in the picture below, you can see what it would look like. And some examples are mollusks and annelids.
And the final step that we have here is the segmented body, which is when the bodies are divided into multiple segments instead of completely together as one unit. And this allowed for more flexible and fluid movements. as they can move and perform tasks with separate parts at the same time. And a good example of this that you can see is a black ant or ant arthropods in general, how each little section is divided and segmented.